good afternoon. Today we're going to talk about some unusual infectious agents that don't fit in anywhere else, but which I absolutely love, and I can't not talk about them. And we can actually look at them in the framework of a question, which is, what is the minimum genome size to sustain an infectious agent? And a corollary is, could there be an infectious genome without encoding any protein? And finally, could an agent exist without any genome at all? And in fact, we can answer these questions today. Viroids, satellites, and prions are going to tell us answers to these questions. So these are the three unusual infectious agents we're going to talk about. You may wonder why prions are here at all. You'll see. I'll tell you about it later. So let's start with viroids. That is a viroid. It's a circular, single-stranded RNA, no protein coding regions. That's why it's called a viroid. I like that name, viroid. It's not a virus because I guess a virus would have to encode proteins. It's just to indicate that it's something different. And the RNA, as you can see, is highly structured, lots of base pairing, so it's almost like a double-stranded RNA, but there are some bubble regions here. It's a naked RNA in nature. There's no protective coat. It goes from host to host as a naked RNA. No receptors needed, obviously, because the RNA can get into the cell on its own. When this is introduced into plants, which typically occurs by contaminated farm machinery or aphids or other organisms piercing the plant cell and introducing this into it, these replicate. They replicate in plants, and they may cause diseases in plants. So these are agricultural pests, and people work very hard to try and get rid of them. There are two families of viroids. Now remember, all the families of viruses we've talked about so far are viridae. These are viroidae to indicate that they're viroids. So we have viroidae, they replicate in the nucleus, and viroidae replicate in chloroplasts. And we'll see about this in some detail in a moment. So these are anywhere between 120 and 475 nucleotides in length. So they're pretty short. And as I said, the RNA is extensively base paired. It looks like a rod. If you did an electron micrograph of these, they would appear to be rod-like. And some of them have a very interesting feature called a ribozyme, which we'll talk about. This is an RNA enzyme, and it happens to be essential for the replication of those viroids that have it. And I like to see viroids as distinct from viruses because they're parasites in a different way. Viruses are parasites of the host translation machinery. At the lowest level, viruses cannot translate their own messenger RNAs. They also require a lot of other host functions, but at the lowest level, they're parasites of the translational machinery. Viroids, they don't make proteins, so they're not parasites of the translation machinery, but they're parasites of the host transcription machinery. They depend on the host for their replication. And viruses, of course, some of them depend on the host for transcription, but not all of them. So that's why I say viruses, translational parasites, viroids are transcriptional parasites. So we'll see how that works in a moment. Here's the first viroid ever discovered, potato spindle tuber viroid, discovered in 1967, and so it's the prototype for these small nucleic acid-based agents. I think there are about 1,800 or so different viroids that have been described to date. There used to be a database of viroids, and it's offline. Sad. How sad can that be, that the database of viroids is offline and I can't go and look at it to see how many viroids there are? But there used to be one, I think it was about 1,800 different viroids. So this one causes problems in potatoes. And you can see here, these are potato plants that have been infected with viroids of different severity, and you can see the healthy plant. And these viroids cause stunted growth, so they're not good for farmers, obviously. You can't have viroids. And I was just up at Cornell. I have an asterisk here to remind me to tell you. I was just up at Cornell a couple of weeks ago, and I met with this plant virologist who told me an amazing fact. He said, every potato you eat began in a lab. Every potato you eat began in a laboratory, okay? And that's because what farmers do, they plant potatoes, and after 6 or 7 years, they become totally infested with viroids and viruses that are brought to them by aphids and and they're no good anymore for production. So what he does in his lab is he derives potato plants from single cells that are viroid and virus-free, and he propagates them, and then he gives them to a place upstate, and they make tons of shoots or tubers, and they give them to farmers, and they plant them. And there are many, many different kinds of potatoes, right? So they do this for all sorts of them. And so the farmers plant them, they get good yields, and then 6 or 7 years, they have to replace all the shoots as well. So this is one of the reasons why he does this, is to get rid of viroids and other things as well. But just think of that. Every time you eat a potato, a French fry, that's a potato, you know, right? A French fry, it started with a single cell in his or someone else's laboratory. He said globally, this is the case. You can't grow potatoes for more than so many years because they will die out. So some of these viroids are economically important. Some of them don't do anything, so we don't worry about them at all. Here are some of my favorites. Kadang coconut viroid. How could you not like that name? And this causes a lethal disease of coconut palms. So if you like pina coladas, you're not happy about this viroid. Then we have the hop latent viroid. This infects the hop plant, which, as you know, you use to make beer, but this doesn't affect the hop plant, so the beer is unaffected. So this is one of these viroids that doesn't cause any disease. You can leave it in the plant. Then there's apple scar skin viroid. Don't you love the names? They're just descriptive of what they do. And these infect apples, of course. So if you've ever seen an apple in a store that's kind of modeled like this, that's a viroid. It's fine to eat, of course, although you may not want to buy it because you'll think something else is wrong. But now you will know you can buy it. And the person next to you will say, why are you buying that? It's infected. And you can say it's a viroid and it's not going to hurt you. Okay? So what I taught you will last throughout your consumer history. Now, viroid RNAs have functional regions. These have been studied extensively in many labs. They've been mutated to see what parts of these RNAs are important. So you can see, first of all, there are two general kinds of viroids, depending on where they replicate, the nucleus or the chloroplast nucleus, the PSTVddiscovered. And you can see I have labeled on here a conserved region and a region involved in pathogenesis. And of course, that probably means, as you might guess, that you make mutations there and it affects the virulence of these viroids. And this one here, this is peach latent mosaic viroid. Potato, spindle, tuber, and peach latent mosaic. This one has a self-cleaving site, a ribozyme right there, and it's also has extensive secondary structure, as you can see. So how do they replicate? Well, this is the exception to the viruses, RNA viruses not being replicated by the host cell. It's not really an RNA virus. These are copied by host RNA polymerase 2, sorry. So this is, of course, a DNA-dependent RNA polymerase, but it can recognize these viroids and make copies of them. So that's amazing. As I told you, one group of viroids has within it what's called a ribozyme. This is a self-cleaving RNA. And this idea that RNA can catalyze a reaction without protein was discovered in 1981. We know now that the RNA in the ribosome can catalyze protein synthesis without any proteins. We know there are lots of examples of self-splicing introns, or RNAs that can cut themselves out. And this is another one. This is an RNA sequence that can just cut itself open. It's beautiful. And this is going to be used to cut the genome as it replicates, as we'll see in a moment. So there's one set of viroids that have a ribozyme, but not every viroid has a ribozyme. Others are cleaved by host nuclear enzymes. So here is an example of how the viroids replicate. These are replicating in the nucleus. So these viroid RNAs, they're again naked RNAs, they enter the cell by a vector or farm machinery. They go into the nucleus, and there they're copied by Pol2, DNA-dependent RNA polymerase II. Even though this is an RNA template, it can be recognized by Pol2. And remember, all the RNA viruses we've talked about are not recognized by Pol2, so this is very unusual. And this genome is copied into concatemers, which means multiple unit length copies of the genome in linear fashion. First you make an anti-genome and then a genome. I'm not going to call them plus or minus, so that wouldn't make any sense to you. You would say, but wait, I thought plus means translatable, and you would be right. So that's why we don't call them that. We call them genome and anti-genome. And then they get imported into the nucleus. They're much longer than this. They're not just 2 units, they're multiple, but for simplicity we show 2. And then in the nucleus, cellular enzymes cut them into single unit lengths, which then circularize. They get exported out of the nucleolus, out of the nucleus, and then they can go from cell to cell through the plasmodesmata. You know, the plant cells are connected by these passages, and so these will then move into every cell and infect the whole plant. So that's how these nuclear replicating viroids multiply. The avsun viroids multiply in chloroplasts. And again, these are the ones that have a ribozyme in them. They come in, they enter the chloroplast, and then they seem to be copied by a DNA polymerase that's present in the chloroplast. And so you have copying, again, of unit-length multimeric RNAs, which are cleaved by the self-cleaving activity of the viroid, the ribozyme. So again, this is not cleaved by cell enzymes. This is cleaved by, it's a self-cleaving activity of the viroid itself. These are then copied to form genome, and again, multimers, which are then cleaved. And then these new products go out of the chloroplast and move to adjacent cells. And again, these can spread throughout the entire host. So the differences between these two kinds of viroids, where they multiply, what kind of enzyme copies them, it's a different enzyme in each case, and how they're cleaved, how the multimers are cleaved, once by the cell and once by the ribozyme. So where do they come from? Well, in the old sense, they were probably the relics of the RNA world, right? So remember, there was first a time on Earth when RNA molecules formed in the solutions on the Earth's surface. They began to replicate, and probably some of them were viroids. And the ribozyme was needed to release genome length pieces of viroids, and that's the ribozymes originated back then. And so I think these have been with us ever since. When cells arose, I think these entered cells because it possibly was more efficient to replicate in a cell and so forth, and they've been in cells ever since. But in modern times, we think they originated in the 20th century. You know, all the crops that we grow now are grown from genetically designed lines. They're monocultures, so you know all the corn in one country is pretty much the same, made by the same company. And so we think when those lines were being developed many years ago in the 20th century, there was just a chance transfer from wild plants to these crops. The wild plants had viroids in them. They probably have for years, and so they were introduced into crops, and now it's very difficult to get rid of them. I think you'd have to synchronously wipe out all the crops in the world and then introduce new lines, but you still probably wouldn't get rid of them because aphids will feed on wild plants and pick up viroids and reintroduce them. So insects certainly introduce these into plants, but there's also mechanical transmission by contaminated farm machinery, hands, equipment from plant to plant, and so it's very difficult to contain this. The fellow up at Cornell who I talked to, he's trying to do similar things with grapes that are used for wine making. They also have problems of viruses. And you know, he said you can't just tell a farmer, here, take this grape and use it to make wine, because they have historically had many different kinds of grapes that they use to make wine, and it tastes good. They don't want to change it. And they often will go to their neighbor and get cuttings from their vineyard. And so he said, you can't control contamination at all. You can't just say breed from this one line because they won't do it. So how do they cause disease is an interesting question. So as I've said, these can cause stunting of plants, which makes it very difficult to have a good yield. The way we think it happens is really interesting because they don't encode any protein. And for many years, no one had a clue how they would cause disease. And then small interfering RNAs were discovered, and people thought, aha, this must be it. And so people did research on it. And it turns out that when viroids come into a plant cell, the plant cell chops up the viroid RNA into small RNAs, 21 to 24 nucleotides in length. And these then silence host genes. They silence genes of the plant, and that's what makes the plant sick. So in some cases, it's not good for the plant. Other cases, it's benign. It doesn't really matter. It's not clear why the viroids do this. You would think that there must be some benefit for the viroid, but no one sorted it out, because obviously there's some that can propagate with or without causing disease in plants. So it's quite an interesting mechanism. And so you can show, and people have shown in the lab, that symptom development correlates with the production of these small RNAs. And many of them map to these areas where I've labeled pathogenesis. So people have done over the years mutations here that reduce the pathogenicity of these viroids. It turns out that many of the siRNA map to these regions as well. So small interfering RNAs derived from the viroids silence plant genes, and that's what causes disease. All right, so that's viroids. Very, very neat things. The next topic, satellites. These are, well, some people think these should be viruses. I'll tell you a little bit about that controversy in a moment. But these are agents, let's just call them an agent for now, with single-stranded RNA, DNA, or circular RNA genomes, a variety of genome types. And what they all have in common is they can't replicate without a helper virus co-infecting the cell in which they are present, all right? And so basically they lack the genes that are needed for virus replication. They can't do it on their own. And there are two kinds of satellites. There are satellite viruses, where the satellite genome actually encodes structural proteins that encapsulate the genome, so they make different particles from the helper. They still need a helper to do other things, but they can encapsulate their own genome. So we call them satellite viruses because they can make a particle. And then we have satellite RNAs, which don't encode capsid proteins. They're packaged by helper virus proteins. They also need other helper virus proteins in order to replicate. Some of these don't encode any protein, actually, but they are packaged in the helper virus capsid. So you can see in this diagram here, there's the helper RNA and the satellite RNA. So these are different from viroids. The ones that don't code for protein are still different from viroids because they get packaged into virus particles, where viroids are not packaged into virus particles. So those are satellites. Now, we've talked a little bit about one kind of animal virus satellite, and that is adeno-associated virus, which is a parvovirus. These are the viruses with single-stranded DNA genomes with the two T structures at either end. We've talked about how they replicate using the T structures as primers. Well, not all parvoviruses need a helper, but there is a certain subset of helper-dependent parvoviruses, and this is one of them, adeno-associated virus. This is going to show up next time. We actually mentioned it very briefly in the last lecture. It's a great vector for gene therapy because you can easily manipulate it, and when you infect people with it, it lasts forever. It seems to last a very long time. So we'll talk more about that next time. But this needs adenovirus to infect the same cell in order for it to multiply. This AAV encodes its own capsid proteins, and of course its DNA gets into the nucleus and gets replicated. It does need to make an mRNA encoding a protein that's needed to drive viral replication, the Rep7868 protein, but it will not complete a replicative cycle without helper functions from adenovirus, and these include the E1A proteins and others that are needed for efficient transcription of the AAV genome. So this will happen only in a cell infected with adenovirus. But these viruses replicate their genomes and they get packaged into capsids that are encoded by the helper genome. They just cannot carry out the entire replication cycle without adenovirus. Now, there are also satellites of plants. Again, these are satellite viruses that are defective. They need another virus to complement them, and so they can encode their own capsids or be packaged in the host. These cause distinct disease symptoms that are not seen with the helper virus alone. So the helper virus alone in the plant causes one kind of disease. With the satellite virus and the helper together, you get a different disease symptom. And that's shown here. This is chlorosis, where you have a mottled coloring, which is due to the lack of chlorophyll in parts of the leaf. And necrosis, of course, is destruction of the plant cells. And these are distinct viruses. They are not derived from the helper virus. It's a totally different genome sequence. They have no homology with the helper whatsoever. So somehow, over time, these have evolved to be dependent on the helper virus for their replication. So here are some examples of satellite viruses of 3 different hosts. We have vertebrate satellite virus, adeno-associated virus, which I've just told you about, which needs a helper of adeno, or herpes. Herpes virus can also serve as a helper for adeno-associated virus, which make their own particles and encode their own capsid proteins. Then we have some other animal satellite virus. So the helper is chronic B paralysis virus, and the satellite is simply called chronic B paralysis virus satellite. This is an RNA virus that encodes a capsid protein. And then we have a plant one. Tobacco necrosis virus is the helper, and the satellite is called tobacco necrosis virus satellite. So again, the presence of the satellite makes a different disease for these, the bee and the plant virus, if you will. Now there is a single human virus that looks a lot like both viroids and satellites. It has properties of both, and that's hepatitis delta virus. So as you will see, the genome looks like a viroid, but it encodes a protein and it's packaged by a helper virus, so it looks like a satellite as well. The helper virus is hepatitis B virus, and so this will only infect people who are infected with hepatitis B virus. You cannot have a hepatitis delta virus infection on its own. And it's thought to make the disease of hep B more serious in the presence of delta virus. And here's the global distribution of hep delta. You know, there are 350 million carriers of hepatitis B virus, and about 5% of those are also infected with hepatitis delta virus. So how would you get this? Well, remember, hepatitis B is transferred by blood products or intravenous drug use. So you could have hepatitis B already and then get another dose that includes hepatitis delta, so you could be super infected, or you can initially get both hep B and hepatitis delta at the same time. And you can see the global incidence here. Very high is black here, and no data white, but you can see certain parts of Asia Pacific region have very high incidence of this as well. There used to be higher in Europe, and it's been declining there over the years. Now here's the genome of hepatitis delta virus at the top. The genome is a 1.7 kilobase RNA. It's circular and fully base paired. So it doesn't have any bubble regions like the viroids, but that looks very much like a viroid. So that's why I say it has some properties of the viroid. But it does encode a protein. So there's an antigenome made in infected cells. And this not only encodes a protein which is called delta antigen. There are actually 2 forms of delta antigen made from a single mRNA. So the mRNA is the same polarity as the antigenome. So clearly the mRNA is either derived from the antigenome, and that happens to be the case, or would be copied from the genome. And as I said, there are 2 different forms of the delta antigen. So it's not viroid-like because it encodes protein. And that's one of the differences. So 1.7 kilobase. It also has a ribozyme activity, which is this little red dot labeled self-cleavage. So that sequence can just cut itself. It folds in a way so it cuts itself and linearizes itself, very much like the viroids. And so hepatitis delta is packaged in the capsid of hepatitis B virus. So here's hepatitis B virus, which is the helper. We've seen this many times before. It's the gap double-stranded DNA genome in a protein capsid, which is surrounded by a membrane, and then their viral protein, the hepatitis B surface antigen, is embedded in the membrane. They're large and medium and small versions of it. And here on the left is the delta particle. So again, it's the membrane and the proteins of the hepatitis B virus with the genome of delta inside of it. So there's no hep B genome in this, just the delta genome. And the large delta antigen and the small delta antigen are thought to coordinate the presence of the delta genome in the particle in this way. So you can see the blue one, the blue protein put in the membrane is large delta antigen. And it's thought that this is attached to the delta RNA. And then the delta RNA in turn is coated with small delta antigen. So that's what those two antigens do. The large and the small are part of the structural makeup of the particle. You could say that the small delta antigen makes up a ribonuclear protein if you want. Now when this RNA is delivered into cells, so the hep B capsid binds receptors, it delivers the genome into the cell just like it would be delivering the hep B genome, except this is hepatitis delta. Here's the minus strand genome that's taken into the cell. It's copied by RNA polymerase II. So this was another surprise. It took years to figure this out because no one knew what was copying this. It doesn't encode its own polymerase, but again, it's an RNA molecule, very much like viroids, that's copied by host RNA polymerase II. And the polymerase simply copies that genome, makes antigenomes, and here you can see in green, we're making multiple unit length antigenomes. And these can be cleaved at the red circle, which is the ribozyme self-cleavage site. And that cleavage can produce an mRNA, in green here, polyadenylated mRNA, which can give rise to the delta antigen. That requires a different kind of processing from making full-length antigenomes, so that's shown here. If you cleave from ribozyme to ribozyme, you get a full-length genome, antigenome, which then circularizes it, and now you have a full-length antigenome which has been copied from the minus strand input. Then that can be copied again to make the actual genome, which is shown in the olive green color. And again, the ribozyme will result in the liberation of unit length copies which circularize and then can go on to be packaged. This is a hepatitis B virus infected cell. It will become packaged. If it's not hep B infected, the RNA will replicate, but it won't be packaged, and that'll be the end of the infection. So the ribozyme is very much like a viroid strategy, the copying by RNA pol II, but being a helper, having a helper virus and encoding a protein makes it more like a satellite. So that's quite an interesting virus itself. Again, it's the only mammalian virus like that we know of. There may be others out there we haven't found. It's the only one with this viroid satellite-like appearance. Now, another class of satellites is what we call virophages. So when they were first discovered, they were discovered by a French group, and they called them virophages, which I think is an unfortunate name. The name comes from phage. Well, phage itself, bacteriophage, is from Greek to eat, because they were originally thought to be destroying bacteria, which of course some of them can do. A virophage is a virus eater. And these are circular double-stranded DNA virus with an icosahedral capsid that only replicate in cells that are infected with a giant virus. So they're satellites, they need a helper virus, but the helper virus in this case is a giant virus. And these particular satellites tend to be pretty big compared to the other satellites that we know of. And they typically interfere with replication of the helper virus. So the other satellites typically don't do that. But these not only benefit from the helper, but they decrease their yield, and that's why they're called virophages, because they reduce the replication of the giant virus. And so here are some examples of virophages that have been discovered. So the first one was Sputnik virophage. It was discovered in a cooling tower in Paris, and its helper is mimivirus, the first giant virus that was identified. And you can see it's got an 18,000 base pair double-stranded DNA genome and encodes 21 proteins. So these are pretty complicated viruses. Then you have a bunch of others. Here's Ma virus, identified off Texas, which is helped by the Cafeteria rowenbergensis virus. And Cafeteria rowenbergensis is a marine phagotrophic flagellate. This is a very common eukaryote in the oceans that tends to eat lots of other things. That's why they call it cafeteria, because it eats anything. Not funny? I didn't make it up. I didn't make it up. And you can see it's big as well. And then there's a number of others here. I like this one. From a contact lens fluid of a patient with keratitis. They called it lentille, which means lens in French. And then a bunch from Yellowstone Lake, where the hosts are not quite known, and so forth. So there are quite a few. And in fact, if you look at the sequence databases from different parts of the world, you can actually find what look like virophage sequences all over the world. Mostly it seems to be in the ocean, but that may be a result of having lots of data sets from the ocean, but there's some on land as well. So there are lots of virophages out there. They do a lot of very interesting things. So here's the structure of one of them. You can see it's a complex icosahedral capsid. And here on the left are some electron micrographs of what these virophages do to cells infected with a giant virus. So this is a mimivirus infected cell. And on the upper left, panel A, these big particles are mimiviruses. And this dark thing, this dark structure is a mimivirus factory. These viruses replicate in the cytoplasm of the cell in what are called factories. And that's what this comes from. And you can see in this factory are smaller particles, right, with the white arrows. Those are the Sputnik virophages. They will only replicate in cells infected with mimivirus. As you can see, this mimivirus at the bottom here has the typical filamentous halo around it, which we saw before. Now, these, as I said, the Sputnik virophage messes up the replication of its helper. It's kind of a nasty thing, right? Here you are taking something from a virus and you're messing it up, but that's the way it's evolved. And the rest of the panels here are examples of that. Panel B, some of the mimiviruses have Sputnik viruses in them. So that obviously makes them defective instead of a genome. You can see that also in C. And then in panel D, look at this. There's this really aberrant outer structure on the mimivirus particle, which has again been caused by the presence of the Sputnik. You can see it in panel E and in panel F as well. So lots of aberrations caused by Sputnik mimivirus. Another virophage is called Ma virus. As I showed you, it's a virophage of a virus that infects Cafeteria rowenbergensis. So this little guy up at the upper right here is Cafeteria rowenbergensis. It's a marine flagellated eukaryote that eats lots of other things. And there are just tons and tons of these, and there happens to be a giant virus of it, and that giant virus has a virophage that infects it. There's a virophage of viruses that infect algae. These are freshwater organisms. And so a number of people have been studying these to figure out what they do. It looks like one of the things they do is exchange genes between viruses. They can find virophage genes both in giant viruses and in host cells. So you must know about the phenomenon of horizontal or lateral gene transfer between organisms. Well, these virophages may be facilitators of that. They also have impacts on ecology of the ocean because they can protect certain cell populations from being killed by the giant viruses. So they can have some effects on ocean ecology. And not many people study these, but I think they're going to turn out to be rather important. Now, the people who study these are offended that they're called satellites. I've been to meetings where there are huge arguments about why they should not be called a satellite. And here's the crux of the argument. You know, many of the DNA viruses we have talked about in this course depend on the transcriptional machinery of the host, right? They don't have their own RNA, DNA-dependent RNA polymerase, for example. They don't have transcription proteins. We don't call those satellites, right? The only difference between them and the virophages, except they depend on transcriptional machinery of another virus, which brings it into a host cell and establishes it. So, right, both viruses, the satellites, the virophages, and the conventional viruses we've talked about, they depend on a transcriptional machinery, and it's just the origin that's different. One is the cell and the other is the virus. So for this reason, they say they shouldn't be called satellites, they should be just called viruses. You know, we call adeno-associated virus a virus. It's helper dependent, but we don't call it a satellite. So this is slowly being recognized that these are probably viruses, not just satellites. And so that's going to change. But I think it's an interesting argument because it's absolutely correct that it's just a dependence on transcriptional machinery. All right, let's spend the rest of the time talking about prions. You could say prions or prions, doesn't matter because it's a fabricated word. So what does it matter how you pronounce it, right? These are infectious proteins that have no nucleic acid. They move from organism to organism without a genome. It's just the protein that causes disease. And these are often in the news because people are fascinated and scared by them, as you will see. I found this picture, which I think is great, Prion Road. Not sure where it is. Would love to live there. Prion Road with my virus's license plate, that would be pretty neat. Mad cow disease was in the news. It's actually often in the news. We'll talk about that. Creutzfeldt-Jakob disease, Scrapie, Kuru, chronic wasting disease of deer and elk now is being more and more written about. 1997 Nobel Prize in Medicine went to Stanley Prusiner for discovering these. And of course, probably the most famous death by Creutzfeldt-Jakob is George Balanchine. This is a New York Times article in which, he was the director of the New York City Ballet, and he died mysteriously, and then an autopsy at Columbia Presbyterian revealed that he had Creutzfeldt-Jakob. And this was in 1983, when we had no way to diagnose this affliction until the person died and we could get specimens from the nervous system. Now we can get some idea ahead of time what's going on, as you can see. So these prions cause what we call transmissible spongiform encephalopathies. Right? So transmissible because they can go from person to person or animal to person or whatever. Encephalopathy is simply a disease of the brain, and spongiform because when you take sections of the brain and look at it, it looks like a sponge. They have holes in it that should not be there. And these are agents of they're uniformly fatal neurodegenerative disorders of mammals, not just people, but other animals as well, as you will see. There are thousands of humans diagnosed each year with prion TSEs. About 1% of those arise by infection, and the rest are either genetic or sporadic, and we'll talk about those. And as recently as 2002, a number of people had contracted Creutzfeldt-Jakob from eating meat from animals with BSE. All right, it doesn't matter how well you cook your hamburger. You can't inactivate a prion with autoclaving or heat. So even well-done hamburgers, you could contract BSE. So you're out of luck. Just have to be careful where you get those hamburgers from, but we'll talk about that in a moment. There are a number of TSE diseases of animals, non-human animals, and they're listed here. BSE, bovine spongiform encephalopathy, that's probably the best known one. Mad cow disease, it was called. We'll talk about that. Then we have chronic wasting disease of deer, elk, and moose, and this has been in the news a lot. We'll talk about that. Then we have exotic ungulate encephalopathy of Nyala and greater kudu. Cats can get this. Your cat at home, your pet, or lions in the zoo, they can get it, feline. Sheep and goats get Scrapie, and mink can get it as well, transmissible mink encephalopathy. In humans, we have a number of diseases that we recognize. Creutzfeldt-Jakob CJD. Then we have fatal familial insomnia. Okay, fatal familial insomnia, which crops up now and then in the press, Gerstmann-Straussler syndrome, Kuru, and then variant CJD, which is caused by eating beef that's contaminated with mad cow prions. And these are all forms of TSEs that are slightly different. That's why they're given a different name. So we call them spongiform encephalopathies because the infected brain has holes throughout it. As I said, this is a section from the brain of a sheep with Scrapie, which is the name for the TSE of sheep. And these holes should not be there. It should be a nice, smooth, filled-in sheet of neurons and supporting cells, and these holes are aberrant. And as this disease develops, the humans or the animals develop psychomotor dysfunctions. They develop motor dysfunctions as well, and cognitive disorders. The exact symptoms depend on which part of the brain is damaged, and every disease, all the animal and human diseases that I listed, have its own characteristic symptoms and pathology, and that's why they have different names as well. So the first TSE that was recognized was Scrapie. And this has been around for 250 years in European sheep, at least we've known about it. It's called Scrapie because the infected sheep rub themselves on fences and then they rub off their hair or fur, I guess. And there's a sheep with Scrapie. You can see the hindquarters has been rubbed clean. And these animals have motor disturbances. They tremble uncontrollably. So in Canada, this was called tremblement du mouton because of that. You know, they're just trembling. They get paralyzed. They have weight loss and they die. So this is obviously not good for sheep farmers. But European sheep farmers did recognize it and they learned that it was infectious agent. In some countries, we still have it. In the UK, 1% of the sheep come down with Scrapie every year. I would not eat mutton, frankly, in the UK. That's what sheep is, mutton, right? Because you might get Scrapie from them. Although I don't know any direct Scrapie from sheep, so maybe it's okay. So the farmers noticed that their animals could transmit Scrapie to healthy herds. They would bring a sheep to the neighboring farm and would develop Scrapie, and then that farm would develop animals with Scrapie. So they were smart. They were like the early epidemiologists. They figured out it was an infectious agent. In 1939, it was shown that if you took the brain of a Scrapie sheep and ground it up and filtered it, the infectious agent went through the filter. So they would take the filtrate and inoculate new sheep, and it went through the filter. So they said, wow, it must be a virus of some kind, which makes sense, right? A filterable agent. But it turned out to have very unusual properties. First of all, it was highly resistant to UV inactivation, more so than any virus that had ever been seen. It was resistant to ionizing radiation. It was resistant to formaldehyde. And finally, nobody could find any nucleic acid in it. And this debate went on for years. I mean, people were saying, oh, you don't know how to purify Scrapie. You're losing the nucleic acid. There has to be some nucleic acid in it. And this went on and on and on. Until it was eventually shown to be a protein by other ways, which I'll tell you about in a moment. This is an early experiment, 1984, where it's basically an inactivation curve where we've taken different viruses and we have given it different doses of ionizing radiation from 10 to the 4 to 10 to the 7 rads. And we just ask, how much do you need to inactivate infectivity? And we have on the left the molecular weight of the nucleic acids here, and you can see that there are two distinct curves. There is a curve for DNA viruses. So here at the upper left, these are the DNA viruses with big genomes like herpes and some of the T phages, adenovirus. It's really easy to inactivate them because the genome is big. We say it has a large target size. It doesn't take a lot of UV to get one hit which will inactivate infectivity. But the smaller genomes like SV40, polyoma, you need more you need more ionizing radiation to inactivate their infectivity. And the same is with the curve with RNA viruses. The bigger genomes need more ionizing radiation. The smaller ones need less. But here is the Scrapie agent. So when they subject the Scrapie agent, whatever it is, so they take sheep with Scrapie, grind up their brains, show that that can transfer, filtered material can transfer infectivity. Then they inactivate it with ionizing radiation and then infect sheep. It takes a lot of UV, more than any virus pretty much, to inactivate infectivity. Now these are pretty hard experiments because the animal is a sheep. Your assay is getting a sheep sick, and it takes a long time. It takes months to a year in order for that to happen. So these are very arduous experiments that only a few people wanted to do for known reasons. But people began to study animal TSEs like Scrapie. They were eventually introduced into other animal species as well. And at the same time, of course, we had a history of human TSEs for quite a few years. And it turned out that the animal and human TSEs had a lot of similar properties, histochemical abnormalities, plasma membrane problems, vacuolization, holes in the CNS, loss of neurons, spongiform appearance, as I showed you, accumulation of glial fibrillary acidic protein, and amyloidosis, the accumulation of fibrils. Now, this is common in many neurodegenerative diseases, like Alzheimer's and others, and the protein that is accumulating is different. This is the prion protein, which is actually accumulating in this case. So human and animal TSEs share these. So as I say, the way these agents were studied is by grinding up an organ, the brain of a sheep, for example, filtering it and put it into susceptible species, and these develop disease. In humans, the neurological degeneration takes many months or years to develop. The agent, depending on how it's acquired, and you'll see the different routes in a moment, it can spread to the CNS from peripheral sites, or it can accumulate directly in the CNS. In the CNS, the protein accumulates, and you get the pathological changes accumulating, and that, of course, leads to the disease. And it's important to remember that there's no inflammation, there's no antibody response, there's no cellular response. This is, as you will see, it's our protein that is misfolding. So we don't recognize it as foreign, and there's no response to it, except the pathological response. So in general, these diseases in people are undetected before symptoms develop. Now, we have now developed some blood assays, which I'll tell you about in a moment, but that's only in the past year or so. So for the most part, the only way you knew was to have certain kinds of symptoms. They're not treatable. We have no way, even if you are diagnosed, we have no way to treat you, and these are invariably fatal diseases. So they're rare, but you're going to die from them. There's no way that we can take care of them. Now, in 1967, Griffith, who was a mathematician, he looked at all the data and he said, this has got to be a protein that's causing these diseases. And people didn't like that because no one could understand how a protein could cause disease. But Stan Prusiner, who eventually got the Nobel Prize for his work on this, he, in 1991, was able to purify the infectious protein from sheep brain. So remember, he would take a sheep with Scrapie, take the brain out, homogenize it, filter it, and then take the filtrate and try and purify the active agent. And he was able to do that, and he purified a protein which he could transmit to other animals, to other sheep, and cause disease. So he coined the word prion, which stands for proteinaceous and infectious particle, prion. I think it's a good name. I like it. And then, of course, when cloning and recombinant DNA became easy to do, the gene for this was identified. The gene was called PRNP. And as you'll see, this is essential for the pathogenesis of TSEs. If you don't have the PRNP gene, all people have this gene, but you can experimentally remove it from animals, they will not develop a TSE. So our current view is that we have a normal host protein called PRPC. Prion protein C stands for cellular. And this is found on the outer surface of neurons. Not sure quite what it does. If you knock the gene out in mice, they're fine. They're not clear what it's doing at all. And under some conditions, this misfolds and gives rise to what we call an abnormal conformer. If you give this misfolded protein into another animal, so if you take the Scrapie protein from a diseased animal, this protein is misfolded, and you put it in a new animal, it doesn't replicate on its own. It can't, there's no nucleic acid, but what it does is cause your normal protein to misfold. And the more you get of misfolded proteins, they cause other normal proteins to misfold, and it goes on and on and on. And that's how you accumulate misfolded protein, and eventually you develop the disease. So the normal conformer is called PRPC, and the abnormal conformation is called PRPSC. SC stands for Scrapie, just in honor, I guess, of Scrapie, the original organism in which this was studied, but we call all the misfolded forms, no matter what the origin, PRPSC. So here's the biochemical basis of that. We have the structure actually of PRPC has been solved, and it consists of two alpha helices and a bit of beta strand here, two beta sheets, two beta strands. The abnormal protein, PRPSC, has a different conformation, has lots of beta strands. It has higher content of beta strands. The structure of this has only been solved recently with much difficulty because it's a very hard protein to work with. It precipitates, it doesn't behave properly. We think the main difference is that it folds differently. So again, it's the same protein. It hasn't changed at all. It's just misfolded. And in this state, it can catalyze the misfolding of other normal cellular prions. It's an amazing quality of that protein. Now here on the left is an assay that you can use and was used for many years, still is, to distinguish the normal from the misfolded protein. And that's digestion with a protease called proteinase K. And if you take the normal protein, PRPC, and digest it with proteinase K, it will digest it to completion. And if you run this on a gel and do a Western blot with an antibody, you don't see anything. But if you take the conformationally altered PRPSC and digest it with proteinase K, it's mainly resistant to digestion. You only digest off the N-terminus, and you have a C-terminal fragment, which is diagnostic. You can take this and run a Western blot, and you will see this protein about 27 to 30 kilodaltons. And so if someone has died of a suspected prion disease, you will take an extract of their brain and do a Western blot. If you see this, confirms the diagnosis. And that's how they will do it nowadays. So it's a good diagnostic test. However, it's obviously not good if you are developing symptoms of a TSC and you want to know if you have a TSC. They can't take a bit of your brain to do a Western blot, okay? It's not really, well, if you want to volunteer it, I guess you could, but it doesn't get done that way. So there are other assays which I'll tell you about. So mice, once the gene was identified, you could make knockout mice lacking both copies, and they're now resistant to infection. So we obviously have been able to generate a prion disease in mice, and if you knock out the gene, they're resistant. And that makes sense, because if you introduce a misfolded prion into the mice, what it does is convert all the endogenous normal protein. And if there's no endogenous normal protein because you've knocked out the gene, they can't develop a prion or a TSE. As we'll see in a moment, you can introduce misfolded prions. So in sheep or mice, you can inject them into the animal. You can eat them, so you eat contaminated meat. There are a variety of ways to get it. Or they can be produced by rare mutations. So it turns out that some people have mutations in their PRNP gene, which predispose them to the PRNPs, just spontaneously misfolding at some point in their lifetime. So whether you get the prion by acquiring it, by infection, or by spontaneous misfolding, it ends up accumulating. The misfolded protein accumulates in your CNS. So again, the little bit of misfolded protein causes all the other normal proteins to misfold, accumulates, and when you get a certain level, you lose your CNS function, and eventually you will die. So that's how the gene works. So there are three kinds of TSEs. Spongiform encephalopathies, because they're not all transmissible. They're shown here on this slide. We have the normal PRPC and the misfolded PRPSC. We have an infectious or transmissible spongiform encephalopathy. So that's the top one here. You eat a hamburger that has a misfolded prion in it, and you acquire it that way, or you get a corneal transplant from a person who was developing a TSE, but they didn't know it at the time when they donated their cornea. Lots of examples of that, or blood products, unfortunately. So that's infectious. And we have familial, where you have a mutation in your prion gene that predisposes it to misfolding. At some point in your life, it will just misfold. And then there's sporadic. And these are people who just develop CJ, Creutzfeldt-Jakob. They don't have any diagnostic mutations. The protein just, for some reason, misfolds. And once you get a little bit of misfolded prion protein, that then catalyzes the misfolding of all your other protein as well. So 3 different ways: infectious, familial or genetic, and spontaneous or sporadic. All 3 you can take and transmit them to animals, okay, by inoculation or feeding, because they're all in the end resulting in misfolded pathogenic prions. So let's talk a little bit about human TSCs. They occur in all forms. They can be infectious or transmissible. There's one example we'll talk about in a minute called Kuru. There's also spread by infected corneas, hormones. People used to prepare hormones from blood. We don't do that anymore. Of course, we can make recombinant hormones, but if the hormone is from a person with a TSE, you're going to give the recipient TSE transfusions and so forth. And often a common one was going to an eye doctor, and they put those things on your eye to measure, right? You ever have that done? You can't stand it. They put the metal right on your eye. Well, they autoclave them, but that doesn't get rid of the prion. And they were transmitted to a number of people by ophthalmologic procedures. Okay, you're not going to go out anymore, right? You're not going to eat. You're going to just stay at home. And we have BSE, another human TSE, which we get from cattle who were fed sick animals. We'll talk about that. And that led to variant Creutzfeldt-Jakob, which was a very different disease from traditional Creutzfeldt-Jakob, which was gotten from eating this BSE beef. So Kuru was a fatal encephalopathy found in the 4 people of New Guinea here. That's Australia down there. This was studied by a number of people, including the virologist Carlton Gajicek. Long incubation period. So he went down there to figure out what was going on here. And it turns out that it was being spread among this tribe because when someone died, they would take the brain out and feed it to the women and children. It was part of the ritual. So the women and children were getting wiped out by this disease, Kuru, which was basically a fatal TSE. So Gajicek went down there and he convinced them not to do this anymore, and that was the end of the disease. So there's no more Kuru in the 4 people of New Guinea. So it was transmitted by cannibalism. So it's not a good practice. And it probably started years ago with a spontaneous TSE that just arose in a person. That person died, and they started eating the brain, and that spread it to people. And then all those people developed it, and then their brains were eaten and so forth. You can see how it's amplifying. Sporadic CJ, this is the one where there's no mutation in your PRNP gene. This is 1 to 2 in a million people worldwide. Typically 50 to 70 years of age, and this accounts for the most TSC. 65% of the TSC appears with no warning or epidemiological indications. Probably this is what Balanchine had, normal PRNP genes. And of course, this can be transmitted to others if you give a corneal transplant or some blood to others, and before you know you have the disease, you can transmit it to others. So again, the point here is that no matter how the protein misfolds, it can be transmitted to others. And as I said, Kuru probably started in New Guinea by the first person years ago that developed sporadic, died, and then everyone started eating it and passing it on. Familial is an inherited disease where you have an autosomal dominant mutation in the PRNP gene. We know exactly what kinds of mutations. So you could have your genome sequenced and see if you're at risk. I had 23andMe done on me and my family, and on the app you can see you're not at risk for TSC Creutzfeldt-Jakob. So I'm happy if you'd like to know that, you can spend $100 and give your DNA to 23andMe or any number of other companies that will do that as well. And again, sometimes these people are developing the disease, they haven't got symptoms yet, they donate blood or corneas or whatever, and they transmit it to others. So no matter how the misfolding occurs, it's transmissible. This is the rate of CJ in the US from 79 to 2015. It's about 1 to 2 per million. That's the global rate as well. The blue bars are the total number of deaths from 79 to 2015, you can see 450 to 500 total. This is cumulative, so the number's going up. And the rate is about the same here. The red is the age-adjusted deaths per million people between 1 and 2 here. Now, the way this works, these 3 different ways of transmission or development of the disease, right? Infectious, genetic, and sporadic. The genetic and the sporadic, the protein is just misfolding in your CNS, right? You don't have to acquire it from anyone. And so that initial prion protein misfolds, and that catalyzes the misfolding of all the others. And once enough are misfolded, then you have the disease. If you eat a misfolded prion, if you eat beef that's contaminated with a BSE prion, we think the prion goes into your gut. There are most likely prion proteins on the surface of your gut cells. We think the misfolded prion will catalyze the misfolding of those, and then somehow they make their way to the CNS. Exactly how, we're not sure, but eventually they get there and cause misfolding in CNS and the CNS disease. Now, BSE was a disease observed in cows in the 1980s. It was an epidemic spread, bovine spongiform encephalopathy. It's a form of cannibalism because we were feeding cows the meat of animals that had died of Scrapie. Now, you can nowadays get grass-fed steak, which tastes different, I understand, from other kinds of fed animals, but it takes a long time for a cow to get to size that's big enough with grass. So in many countries, they give them meat. And in a lot of countries, in fact, there's no grass to graze on. So they feed the cow meat, and that gets them to grow very quickly, and they can be brought to market. Well, it turns out that in the 1970s, they changed the way the meat was prepared. So the way they get this meat is they take dead animals and grind them up, make a paste. It's called meat and bone meal. And as I said, they feed it to cows as protein supplements, and including in these animals were sheep with Scrapie, right? They were feeding these to cows. Now, for many years, that didn't cause a problem, but in the 70s, they changed the way this meat preparation was rendered, and apparently before that, it got rid of, the process got rid of the prion, and now it didn't get rid of it anymore. And so the cows started developing a TSE, which was called bovine spongiform encephalopathy. At some point, so basically the Scrapies infect the cows develop BSE, then people eat the cows and they develop Creutzfeldt-Jakob disease. So we recognized this, we stopped, and we went back to the old way of preparing the meat and bone meal, but the fact was we introduced this disease into a lot of people. So here's the epidemiological curve for this. In total, we think about 1 to 2 million cattle were infected. And here's the problem. The incubation time of the disease in cattle is about 5 years, but you slaughter them at 2 or 3 years to make meat, so you don't know that these cows have any disease. Because as you could see from that picture, a cow with BSE at the advanced stages is quite clear, and you would not make meat from that cow to eat. That would be criminal. But in 2 years, you can't really tell. So these are the numbers of bovine spongiform encephalopathy cases in thousands in the cows. That's in orange here, so BSE is in orange. You can see it starts to go up in the 80s. It's a long incubation period, peaks in the early 90s. And then they changed the meat and bone meal right early on when they saw an increase in the number of cases, but there's such a lag that the cases went up and it finally went down again. And finally they decided to completely ban mechanically recovered meat in the cow feed. And then overlapping this is the variant CJ in people. That's the outbreak in people who ate this meat, and they start to get infected. There was a peak, which has now gone way down, of course, because we don't feed cows this preparation anymore. However, we still do see cases of BSE in cattle sporadically in countries that grow cattle, of course. We think these are sporadic. The cows are just going to develop BSE sporadically, just like we do. We're worried about this because of the food supply issues, but we don't check every animal for BSE. Less than 2% of slaughtered cattle are tested. And that's because you have to actually look at the CNS to tell if they have a TSE or not. So more recently, diagnostic tests have been developed where you could, in fact, theoretically screen every cow if you wanted to. And people are trying to get drugs that would block prion accumulation, but that hasn't worked out so well. So here are BSE cases in North America. We've had some here and in Canada. Again, not from feeding cows dead sheep, but because the cows sporadically develop TSE. The blue ones are Canada, and the light blue ones are after, there was a feed ban in Canada as well, coincident with the UK feed ban. So you can see there are a number of, it's just one here, a year. These are sporadic, and there are a couple here in orange in the US as well. So only 24 from 93 to 2015, but if these get into the food supply, it's tragic because people will get infected by them. So far that hasn't happened. There have been a number of interesting recent papers published which I've blogged about, and one concerns a case of prion disease from contaminated beef. This is a man who developed just a few years ago a fatal TSE. It turned out he wasn't living in the UK, but he had lived there 12 years before, and it was the time when BSE was on the rise in cattle, so they figure he must have eaten beef, and eventually it got him. And then there's one here of a fellow, prion contamination in the emergency room. This is a fellow who shot himself, tried to commit suicide. He shot himself in the head, but he survived for a while. He went to the ER, where his brains leaked all over everything, and he died eventually. And then post-mortem, they found out he had TSE. And so meanwhile, TSEs were all over the ER. And you can't autoclave stuff, it doesn't get rid of them. So I had to throw out all the equipment. Tens of thousands of dollars worth of equipment in the ER had to be thrown out. So you can read about that. So there are procedures in place for this. You know, if someone comes in with symptoms that look like this, you have to contain them differently. This is an unusual situation because he had leaking brain material, but it doesn't always happen that way. So we have now actually a test that can be used to detect prions, misfolded prions. It's kind of like PCR for proteins. It's called quaking and shaking assay. So what you do is you make some recombinant PRPC protein, that's PRPC monomers, and then you mix with it a sample that you think has misfolded prion, and it's going to have some oligomers in it. And what will happen is the oligomers of misfolded prions will cause the monomers to aggregate. So basically make longer and longer aggregates, and you incubate them to get longer. Then you sonicate to break them up, and then you can add more monomers, and they'll make longer aggregates of each one. So you go through like 40 or 50 cycles of this, and you get a lot of aggregated protein, which you can then digest with proteinase K and run on a gel on a Western blot, and you can see if it's a misfolded protein. So this was originally developed using nasal brushings because it was found that not only people but cows with BSE accumulate prions in the nasal, the olfactory neuroepithelium. But now, just in the past year, it's been shown to be very sensitive to detect prions in the blood. So now we can take a person who has symptoms of a TSE, take some blood, and do a diagnostic test. Now, that's the good part. The bad part is we can't do anything for them, but at least we know they have a TSE, and they can be cared for in a way that makes them more comfortable. Now, a last consideration here, there is a prion species barrier, and that means that putting protein, a misfolded protein, into the wrong species is often inefficient. So most of the time, the prion that you inoculate into the host, the prion, the misfolded protein, and the host protein should be the same protein. All right, so the sequence of this prion protein causes tropism. And here's an example of that. If you take a hamster prion, misfolded hamster prion, and you inject it into mice, they don't get sick. But if you make a transgenic mouse that has the hamster PRNP gene, and you now put hamster PRPSC into them, they will get sick. So the inoculum and the host protein have to match. However, it's not always the case. Cow prions have a wide host range, so you can infect mice with bovine PRPSC and they get sick, and we know when humans eat bovine PRPSC, they get sick as well. So we say that the bovine PRPSC has a broad host range, and that's why we're worried about BSE, because it can infect not only us but other animals. And we certainly feed cows to other animals as well, our pets and zoo animals and so forth, and we've spread prion diseases to them. Another concern is a prion disease of cervids. Cervids are deer, elk, and moose. It's called chronic wasting disease. And this is on the rise because we're looking for it here from 1965 to 2015. You can see the increase in the number of states with reported cases. Both captive and free-ranging animals have found to be positive. You can see throughout parts of the US, South Korea as well. And this is an example of a deer with a chronic wasting disease. It looks very much like a Scrapie sheep. Very high percentage. Standing herds 90%, 60%, wild cervids up to 15%. So what happens is these deer shed the prions. They shed them on their antlers, nasal secretion, saliva, skin, blood, milk, give birth. The baby has prions in them, urine and feces. And these survive in the environment. They're quite stable, and other animals can eat them, not only deer, but other animals as well. So it can spread to another deer. It's licking the grass or eating something down there. It's got deer prions in it, and they will get infected as well. So this is spreading in deer populations. A paper published a couple of years ago showed that, in fact, if you add prions to grass, the grass will bind the prion, it will take it up, and it will persist in the grass. It will not replicate, but of course, you can then feed it to an animal and show that the prions in the grass will cause a prion disease in those animals. You could even put prions in the soil. They will be taken up by the roots, go into the plant proper, and contaminate the plant. So there's obviously ample opportunity for a deer to shed prions. It'll be taken up into plants and other animals that can come along and eat it. So we're worried about cows eating these, but fortunately, cows don't seem to be susceptible. Now, if you put cervid misfolded prion into mice, there's no disease. Again, if you make the mouse transgenic for cervid PRNP, you do get disease. So that suggests it has a narrow host range. If you take a mouse transgenic for human PRNP and give it cervid misfolded prion, there's no disease. So if this is predictable of what's going to happen in people, this would suggest that we can't get a TSE from eating deer. But I don't think anyone wants to do the experiment and find out, right? Because maybe this isn't predictive for humans. If you inoculate cows intracerebrally with deer misfolded prion, you will get disease, but if you feed cows, they don't get sick. So this probably means that our food supply in cows are safe from deer prions. However, cows, of course, will spontaneously misfold anyway, so that's a problem. So could prions transmit to cattle? Probably not, because they don't get susceptible by feeding. However, if you hunt deer, if any of you hunt deer and get pleasure out of that and eat the deer, be careful. There's this wonderful site, CWD-info.org, where they tell you how to take care of your deer that you've killed so you will avoid getting a prion disease. And basically, if the deer is acting weird, don't eat it. And, use gloves, avoid the spinal cord. Don't eat the brain, spinal cord, eyes, spleen, tonsils, or lymph nodes. And of course, if the animal is positive, I don't know who would be eating a positive animal. I guess you could catch a deer and bring a sample into a lab and get it tested and then throw it away. Anyway, these are interesting guidelines. So let me end up with a slide that I think summarizes all the different ways these TSEs may have evolved. Maybe Scrapie was the first animal TSE to spontaneously arise in sheep and goats and has been spreading for many, many years from one animal to another. It could be that sheep Scrapie led to chronic wasting disease in cervids, but more recently there's some experimental evidence that suggests that maybe cervid CWD is actually a sporadic disease. These animals are just sporadically developing it, and then they can transmit it to others. We think that feeding dead sheep to a variety of animals has led to TSEs in other species. We talked about BSE. We think transmissible mink encephalopathy may have originated with Scrapie, and BSE certainly has led to TSEs in other cattle and various animals that we feed cow meat to, even domestic cats. There are cases of feeding cow from the British outbreak to domestic cats and causing TSEs as well. Of course, we ate contaminated beef in the 80s and developed variant Creutzfeldt-Jakob, which can be passed on to other people iatrogenically. And then finally, I think humans have also developed sporadic Creutzfeldt-Jakob. The Kuru outbreak is probably a sporadic event, and that can be passed on to other humans iatrogenically as well. So that's prions. Now, why would I have this in a virology course? I don't know. I think it's just cool. Why not? In fact, early on, only the virologists would listen to the people who were studying prions. No one else would listen. So in every virology textbook, that's where the prions would be. So that's why they're here today.